All righty. Okay, so today is the 14th. Do I have updates and things? I do. So the plan right now is we should be back to campus on Monday. At least that's what everybody's saying. Okay. It is entirely possible that that will not happen. But we'll see. So the plan right now is to go ahead and the opportunity session that was supposed to be today will be on Monday. And then the rest of the schedule is just kind of get like bumped down a little bit. I don't predict needing to change any other important dates, like other future opportunity sessions, right? I don't think we're going to mess with any of that. I hate messing with that stuff in general, but I don't think we're going to need to. I think we're just going to need to, kind of crunch a few things in order to make that work. So hopefully there won't be any like long-term, like down the line ramifications about any of that. Elle, are you unmuted because you were going to ask a question or just because you're unmuted?
I just realized I was unmuted. Oh.
Aren't you glad you didn't say anything crazy? I'm always worried in meetings that I'm going to be like, but you know what? Actually, my voice gives it away in meetings when my voice, my face, I like make the, I don't have to swear during meetings because I make the like, anyway. Okay. This is all getting recorded. Anyway, consummate professional, you guys. Okay. Other things, let me share screen so I can show you something. I did finally, with all the drama of, everything getting changed this week, I'm like fell out of my flow, right? And so I finally got the rubric up for the lab report that is due on Friday. So here's the rubric. I'm not meaning for you to carefully read it right now. You can look at it on Canvas. I just want you to see that it's there underneath that assignment. And then, so don't forget that's due Friday by, 11:59 or whatever Canvas's default is on Friday night. So yeah, okay. I also updated the date of the opportunity session in here to reflect that we were moving that, so that in your calendar, sorry, there's a bunch of stuff you don't care about, right? So then in the calendar, it shows that our opportunity session is moving to September 19th. What other updates do I have? One other thing is I did grade, I'm not going to show the grade book to you, but I did grade our chi-square labs. I finished those up. So look at them because there are comments, right? And one of the things that I want to say about that is that the beautiful thing about doing these lab reports, doing several of them, is that I'm not expecting them to be perfect from day one, right? We're all kind of learning how to do this, and it's going to be a process, right? Where each time it's like, oh, you get a little bit better at figuring out how to write a hypothesis, or get a little bit better at how to, interpret statistical analysis or whatever, right? So it's a building process, okay? And so there will be at least 2 more lab reports, something like this, before you all are doing your essentially big lab report, which is your semester project. Okay. So the idea is that these are lower stakes assignments to kind of help you get better for the big one at the end. Okay. So I definitely recommend sort of leaning into the idea that you should be going back and looking at the key. You also should go back and look at, not the key, the rubric, right? And the comments on your actual assignment. I can't talk. The other thing you should go back and look at, which I forgot to show you, is you should look at my lab report that I wrote up for the chi-square. Okay. And I have another one that I will post for the difference in means as well. Right. It wasn't there. it was this week. It was week 3. Okay. Right. So I put in my lab report so that you could kind of see, like, it's think of it as like a key, right? It's like something to compare to. Okay. And so you can see, how, if I was doing this lab report, how I would have done it. Now, in this particular one, you were not expected to do a lab report, right? All you were expected to do is the things that I asked you to do that were essentially components of a lab report. But I just did it as a whole thing just so that you could kind of see it. All right. And so I will always include these when I grade them so that you can kind of see, all right, if she's saying that this wasn't right, where's an example of what does look right? So there's your example. Okay. All right. Let me open the chat and see. Can we have it due Sunday instead? Okay, Maria, here's my concern about that. My concern about that is that I don't want you, I really want you to be studying for the opportunity session too. So I kind of want you to get this done and like off your plate so that then you can spend the weekend working on, studying for the exam. Okay. So I'm not going to move the due date. What I will say is there is a built-in grace period that you can read about in the syllabus. Okay. But I really, I don't want to move it, right? Because I want you guys to get this done so that you can like get, get this out of your way and then carry on. Okay. There was another anonymous question about the teleology assignment. Some of you, you kind of missed the mark on the teleology assignment, which is absolutely fine because this is one of those topics that I want you to keep doing it until you get it right. Right? Because I really want to make sure that we all really understand this because it's a fundamental thing. Okay. And so please, everybody, check your teleology assignment. And if there was a comment in there about, hey, please try this again, I'd be happy to regrade, I will. I would like for you to get it done, but also by the end of this week, just because I don't want that to keep going on and on forever and ever and ever. The way that you can update it is you can either edit your original post or you can make a new post, a new reply. Okay. I made it so that it will let you do that. So I'd like you to get that done by, I think I have it closed on the 23rd. Let me check that assignment. Let's see, just to make sure. Catch a sneaky teleology. How did we do that? So I set it to, nope. I gotta edit so I can see. Sorry, I'm not really editing it. I just need to do that to be able to see the close date. Yeah, so the close date is the 19th. So within 5 days or so. Okay. And if with discussions, I don't automatically see that you have resubmitted. I do with other types of assignments. So for other assignments, I will, you don't need to tell me if you're resubmitting something because it'll notify you. Canvas will tell me, but Canvas doesn't tell me with discussion. So if you are resubmitting, please shoot me a quick message just saying yo, I redid my teleology. Please check it out. Okay. Any other questions about anything else that's going on before we jump into good times and shenanigans? Okay. So it is a little bit, I don't know if convenient is the word, but a little bit not the end of the world that we could not take our opportunity session, because now that allows us to continue with our evolution conversation and have this be on the first opportunity session instead of getting bumped to the second. So as soon as we're done here, I will update the outline based on where we cut off today. But fundamentally, this is kind of a more chill, maybe topic. Because basically now what we're doing is we're just kind of we've baked the cake, right? We have talked about microevolution, we have talked about macroevolution, right? And now we're just putting the little swirly frosting on the top, which is, we understand the processes for how evolution happens, right? Whether it's on a smaller scale or a larger scale. Now let's talk for a minute about the evidence for that. Okay, so some of this I've alluded to kind of as we've gone, and some of it we're going to talk about a little bit. Some of it we cover a lot in 62. So this is going to be kind of like a little bit all over the place. Okay, so the first thing we're going to do is we're going to talk about all of the major, depending on how you count it, there are 4 lines of evidence, right? Types of evidence for evolution. Okay. Okay. And the first one is the physical evidence, right? So the first one is the fossil record. So I want to talk for just a minute about what the fossil record is, and how we use it and that kind of stuff. Okay. So fossil record, one thing that I think a lot of people don't realize is that fossils can be, there are lots of different kinds of fossils, right? So fossils can be, a lot of people think about like bones or shells or hard structures that get fossilized. But a fossil by definition is any like physical sign of life that is left behind, right? So any, there's my bad pen again. It's not the pen, it's the screen. Sign left behind. Okay. So the reason why everybody thinks about bones and shells and all of that kind of stuff is that harder tissues like bones and shells tend to fossilize more easily than soft tissues. But there are still examples of soft tissue fossils, okay? And all kinds of kind of interesting little things. So here's a guy who's standing next to dinosaur footprints, right? So a footprint, even though it's not a part of a dead body, counts as a fossil because it's a, you know, basically a piece of evidence that something was alive and walking. Okay. Insects trapped in amber is another one that everybody thinks about now that, you know, Jurassic Park is a thing. You can even have imprints of structure. So like imprints of leaves or pollen, right? Pollen is a fossil.
Question.
What's up? Yes, ma'am.
So would the recently, or I don't remember when it happened, within the year, they found, why can't I English right now? They found like a woolly mammoth in ice. Would that be considered a fossil?
Well, that's a really good question, actually. I mean, because the tissues are so well preserved, I don't know that you would consider it a fossil or not. Because it's just like it's like something you left in the back of the freezer for too long. Like, I don't know. I think in the strictest sense of the definition, I would say yes. But generally, we're talking about things that are much longer periods of time that, you know, there's no like viable, I shouldn't say no viable genetic material. There's no soft tissue remaining. And the difference with the occasional, it happens regularly where they'll find a woolly mammoth or in some cases they find like really ancient humans in the ice, where it's like they're still, all their, you know, meat is still there. It's just been frozen for tens of thousands of years. So anyway, yeah, so plants can fossilize also, just like animals can. So this is some petrified wood. Petrified wood is a trip because basically what happens is minerals go into the wood, and essentially what you end up with is a rock that looks like wood because the wood was kind of like the pattern with which the rock was made. I have a really impressive piece of petrified wood in my backyard that I'm assuming was found by the previous owners, but it's so heavy that when they moved out, they were like, they just left it there. And that's probably what's going to happen when I move someday. I'm just going to be like, nah, I can't. This is heavy. I don't know where to put this. Anyway, okay. So that's what a fossil is. Fossils are dated a couple of different ways. Fundamentally, the way that we think about fossils in their age is based on this like strata, right? So like layers, right? The older something is, the deeper underground it is, right? The way that a typical fossil bed is formed is you have some sort of body of water, and that body of water is receiving, you have an ocean or a lake or a, whatever, a small sea, right? And it's receiving runoff whenever it rains, right? So things have a tendency to pile up in old lakes and old bodies of water, right? And so that includes dead bodies, right? So dead bodies, it rains, they roll downstream, they end up in the pond, they get covered with muck, and it's this really like anoxic environment, and that slows down decomposition. And wouldn't you know it, it gets covered by more muck and more muck and more muck until there's enough pressure that it starts to, mineralize, yada, 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 right? You end up with something that's preserved. Okay. And so the deeper you go, the older the fossils are. Now, the original, you can see these layers, right? So people have known about this for, this idea of older fossils at the bottom, newer things at the top, for as long as we've been able to like look at geology. Right? Because you can see these layers with which rock was formed. Yeah. And so the original, I'm skipping around a little bit. The original way that fossils were dated is we talked about them collectively, like, there's this layer, and what types of organisms, what types of fossils do we find in this layer? And then there's this layer right above it, and what do we find in that layer? And then there's this layer above it, what do we find in that layer? And that's how the geologic timescale was developed, right? Initially, it didn't necessarily have dates associated with it. It didn't have times associated with it. Or if it did, they were like kind of wild ass guesses, right? Because it's like, who knows initially. Okay. And so it was a matter of, oh, well, we see all of these different kinds of things in rocks of this general, age as far as its depth. Okay. And that's how all these different time periods were named. The fossil record has since, I'll come back to these in a second, right? Has since been, we are now much better at estimating ages of things based on radiometric dating. Okay. So there are a variety of different isotopes that are radioactive. And so radioactivity essentially, I mean, you guys have taken chemistry, so I'm assuming that you know a little bit about this, probably. Maybe. Essentially what radioactivity is when you have atoms that are unstable. And so because they're unstable, they're like shedding energy essentially, right? And that energy you can detect. Okay. And so there's a bunch of different isotopes that are radioactive. The one that we use for more recent fossils is carbon-14, but there are other isotopes that we can use for things that are much older. Okay. Carbon typically has 12, oh my gosh, she's talking about chemistry again. Really? Dang it. Right? Has 12 protons, 12 neutrons, 12 electrons, an atom of carbon. That's why its number is 12. And if you look at its mass, it's like 12 point blah, 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 00 something. Okay. Because most of carbon is carbon 12, right? An isotope of carbon that has 12 neutrons. But there's also carbon 13. And carbon 13 has the same number of protons, the same number of neutrons, but it has one extra, or excuse me, same number of protons, same number of electrons, one extra neutron. Okay. Carbon 14 has 2 extra neutrons. And that makes it, I thought we finished chem talk last semester. No, you're never done with chemistry. And never, ever. So carbon 14, because the nucleus is so big with those extra neutrons, it's not stable. So it like, and it like shoots out particles and crap. It's really, that's very technical chemical explanation, by the way, just so you know. Right. So it breaks down, right? Because it's unstable. And for all radioactive isotopes, they break down at a very reliable pace. Right. So it doesn't vary. It's not like, oh, it's breaking down faster or slower. It's like, it is like you could set a clock to it kind of pace, right? It's very reliable. And so we call the rate of breakdown, the way that we measure it is with half-life, right? So what this little table, don't memorize this, but just, we're looking at it, we're not memorizing, right? So what this is saying that if you have some amount of carbon-14, right? 100, we start off with 100% of whatever that amount is. Okay. Over the course of 5.6 thousand years, so 5,600 years, half of it will disappear. And then another half after another 5,600. You see where this is going? Another half, right? And the beautiful thing about this is it never really, I shouldn't say never. It doesn't go away completely, right? It makes this really nice curve where you're getting smaller and smaller and smaller and smaller amounts over periods of time, right? And so cool. Okay, that's cool. So how does this work for fossils? Well, we can use this to date fossils because we know the half-life is what that's called, right? We know the rate at which it decays, it goes away. And we have an idea of how much carbon-14 exists at any given time, okay? So here's a clam, right? When clams are alive, they are adding carbon to their body tissues, right? So the shell of a carbon, of a carbon, of a clam is made of calcium carbonate, right? And so as it builds its body, it's acquiring calcium and carbonate from the environment, and that carbon is being built into its body tissues. Okay. So, right, if we know that, around the whole planet, approximately, such and such percent of carbon is carbon-14, right? Then what we can do is we can look at how much carbon-14 remains after this animal dies and after it's been sitting around for a while to see how long ago it died, okay? Because at the point that it dies, it stops taking up carbon from the environment, right? So our clam dies, it's covered with sediment, right? It ends up getting compressed and it forms in this, rock, and we call it a fossil, right? But even while that's happening, the carbon-14 decays because of its half-life. And so sometime later, when our friend here with his hammer, her hammer, I don't know, their hammer, okay, let's not be sexist, Sarah, with their hammer, they should have nails like Miguel's picture. Sorry, no, they shouldn't. Not digging up fossils. You'd break your nails. So you would dig up that shell, right, and analyze it to see how much radioactivity it still has, right? How much carbon-14 is in there, and you can say, all right, well, if we know how much carbon-14 there was likely in this guy when it was alive, we can tell you how long it's taken since it died. Okay. Elle asked in the chat if we could do this with humans. Absolutely, right? So we could do this with ancient human bones, right? For sure. You can look at a bone and you can do these calculations to figure out how long ago it died. So any kind of fossil that has any kind of carbon in it, yeah, absolutely. Okay. Right? So like I said, carbon-14 is just one of those things. It's mostly useful for things that are fairly recent in the idea of, the range of 50,000 years or so. So this isn't going to be used for like dinosaurs, right? This is going to be used for humans, right? Or used for more recent, more recent fossils. Okay. But there are tons of different other isotopes that can be used for dating as well. All right. Does that make sense? Here's another picture that shows, so this one is using uranium, right? Which is one that has a slower half-life, right? So this one is useful for, right, really long periods of time. Okay. Yeah. Anyway. Okay. Any questions about dating?
Yes.
Radiometric dating? Yes, ma'am.
So like, okay, you said we can't really do this with dinosaurs since they're so old.
Well, we can use radiometric dating with dinosaurs. We just don't use carbon. We use something, a different isotope like uranium.
Okay, never mind then.
Yeah. So yes, we absolutely do dating of everything all the way back to the beginning, essentially. Right. We just, yeah. Is that okay? We just have to use different isotopes for different things, for different age ranges. Okay.
Cool beans.
All right. Other questions? I'm gonna have a sip of coffee. I'll let you think about what questions you have. Okay. So by doing that, now we're able to slap some dates onto this process. Okay, this list here. So I know this is too small for you guys to like read. You know, it would be if we were projecting it in the classroom anyway. Maybe you have fabulous eyes and you can read this on Zoom. But I don't want you to memorize any of this at all, right? I won't even with 62, right? But you see all these dates here along the sides, right? These were original sort of guesstimates, but then, right, they're like confirmed by radiometric dating. So we know that the sort of boundary between here and here, right, is between 201 and 145 million years ago, right? The reason that wouldn't it be lovely if the chunks were like consistent, like it was like every 50. Why is it not every 50 million years that there's a change in a time period? Why isn't it that way?
Isn't it because you can't set the evolution be it in a specific time frame, so it just goes the way that does? It's not like you can control it.
Exactly, right? So what these were originally based on, these time periods were based on, their names were based on like, what do we see right now? So essentially what these dates reflect is periods of time where there was a, geologically speaking, abrupt shift in what's around, right? So, you know, nobody decided, oh, we should just call 419 million years ago to 359 million years ago the Devonian, right? What happened was they saw a period of time where there was these types of organisms that were in that layer of rock, right? And so, yeah, so it got the name Devonian, and then the dates are added later. Okay, so this is a historical record, not something that humans just developed like how they wanted it to be. And one of the things that I like about this this diagram, and I know it's too small, but you can, you know, go back and look at it later, is it shows big extinctions, right? And so one of the patterns that you'll notice when you look at these is, right, because the time periods are based on what's around then, when extinctions happen, right, that's a new time period. Okay, and what you notice is that following extinctions, you often end up with pretty impressive diversification, right? So here's our first time period, yeah, and so the Cambrian was, you know, this period of time, and then the Ordovician after things started to get kind of exciting, right? And then there was this really big extinction, and a bunch of things died. They disappeared. There aren't fossils for those anymore, right? Because they stopped living, right? But then we get new things. Look at that fish. That's crazy, right? So we end up with these cool new things that we see after each extinction. Okay, so here's a geologic time scale from a different book. It just looks differently, right? I'm not going to talk too much about, well, maybe I will a little tiny bit. Okay, so one other thing I want to say about the fossil record that I think is really important thing to recall is that the fossil record, because it is all accidental and haphazard, it takes really specific conditions for fossils to form. Most things that die are completely broken down, right, by detritivores, decomposers. So it is, if you think about it, right, the number of dinosaurs that actually ended up forming fossils is not representative of all the dinosaurs that ever lived, right? It's a very, very, very small number. And so sort of by definition, we can't expect that the fossil record is going to have examples of every single thing. But what's really interesting is that the fossil record actually does have a lot of really interesting intermediate organisms, right? Intermediate, we call them missing links when they're missing, and then we find them. So these kind of like in between steps. All right. So this particular image is looking at whales. So cetaceans are whales and dolphins, those guys, right? And so what this little tree is showing you is it's saying, okay, here's the ancestor, right? The sort of here's the most close, I can't speak, the closest relative that shared a common ancestor with what we consider to be dolphins and whales now. Okay. And we also have all of these kind of intermediate stages, right? So in a way, it kind of gives us an idea of a progression, right? So first there was this thing that was this hoofed animal that was kind of a little bit like a hippo or a deer, kind of, right? That's the relative of a whale, by the way. Okay. And there was one of the populations that sprung forth from something that looked like that was becoming more and more sort of aquatic. So its legs sort of changed, so there was a little more squat because it was living in shallow water instead of running around on land, right? And so it has all of these skeletal changes that we see that make it more well suited for kind of puttering around in water like a hippo. Okay. And then over time, oh, look, now we're starting to see legs that are definitely looking more like they're flippery, right? We're starting to see skull changes that make it look more like it's spending a lot of time underwater and only just kind of the top of its, yeah. So we're seeing more and more flippers, right? And then we're seeing, oh, snap, hind limbs are gone completely, right? So first they're vestigial, and then they're eventually gone completely. Right? So we can see these intermediate stages. And so when in situations where we have such a beautiful, clear record, this provides incredible evidence of what happened, how things went down, right? We don't always see that, right? So here's another great example of something where we see this incredible record that shows all kinds of intermediate stuff, all right? Now, real quick, come on, pen, work with me. Ignore this for just a second, okay? For all the time that I was learning about organismal biology, right? These were the fossils that we talked about when we learned about the transition from fish to amphibians, right? And it was like, okay, well, we got this fish guy that had these weird bones in its fins, and they're kind of starting to look like limbs, and that's interesting. And then there was this jump from this to this, right? So you don't need to know these names, right? But these are sort of ancient amphibians, and this is an ancient fish, okay? And so the thinking was, well, there has to be some sort of intermediate, right? Something in between 365 million years ago and 385 million years ago, where there was something that was a little bit of a transition between this and this, right? But that fossil had not been found yet, right? People were looking for something like that, but it hadn't been found. And then it was found in, God, I don't have my notes in front of me, so I can't remember. I want to say it was like 2005, right? So fairly recently in the grand scheme of things, right? They did find that missing link, okay? And so I talk about Tiktaalik is his name. I like to talk about Tiktaalik in 62 when we talk about that when we talk about animals, okay? So if you haven't already, you'll hear more about Tiktaalik in 62. But there it is, okay? So there are some stories where we have very clear kind of steps, right? There are some situations where we don't have all of those steps, either because we just haven't found the missing link yet, or because not everything fossilizes. It's a spotty record. It's an incomplete record, okay? So there's Tiktaalik. Yes, Elle.
There's a question in the chat.
Are you going to make me find the chat and read it? What's the question?
I can read it.
That's okay. I got it. Radio ejection. Okay. So no, we're still talking about fossil records. So this is all evidence number one, because all of this is relating to the fossil record. Okay. I saw that. I was actually intentionally not answering. So I was going to say, so that's the fossil record. That's how we use the fossil record. That's how we do dating in the fossil record. Okay. Cool. right? Here's some more, just, for fun. Here's some more, here's another way of viewing our different extinctions. All right. So the one where we lost the dinosaurs. Why? My pen doesn't like me today. The one where we lost the dinosaurs is that one. And that's the one that everybody gets all excited about, right? But it wasn't nearly as big of an extinction as this one, right? That one was major, more than about half of everything that was alive went extinct at that one, right? Whereas the end of the Cretaceous, it was what, like 15% of different living things went. Anyway, okay. And then look, here's the mass extinction that we're participating in right now. That's fun. Sarcasm font. Anyway, okay. All right. Cool. We talked about adaptive radiation before. Okay. So that's our first line of evidence, the fossil record, and then sort of understanding. Maybe I should write that down. I'm not good at writing down when I'm doing Zoom because I'm like, I don't, that's too many things I have to have open. Okay, so the fossil record, can I find it? Can I find a space? Right, so we're not just talking about what is a fossil. So what are they? How are they formed? Right, we're talking about how are they dated? Right, we're talking about missing links. And sort of, how complete the fossil record is, and then extinction. Okay, so that's, the topic sort of underneath that number one, that piece of evidence. Okay. Are we ready for our second line of evidence? Any questions about fossils, fossil record, any of that stuff? I have to tell you guys, for my entire life, one of my favorite things to do when I was a kid was go to natural history museums because I was like a dinosaur nerd kid. That was me, right? I'm that kid. And all that stuff, right? And every time I, talk about the fossil record and, evolution of life, I get all like, like, 7-year-old Sarah is like, wow, 44-year-old Sarah is really cool that she gets to teach people about that stuff. Just saying. Anyway. Okay. Cool. All right. So now on to our second. I'll write a big number 2. Second line of evidence for evolution, dun, 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 is something called biogeography. So what do you think that name means, biogeography? What does that mean?
When you break down the word, it means life of land.
I think. Right. So geography is like where things are. Yeah. So biogeography is, who lives where, essentially. Okay. And so biogeography is really, really interesting, both as like a modern thing that you can study of species that are alive now, but you can also, kind of combine this with looking at fossils to see a different picture. Okay. So everybody's favorite biogeographical example that tells us a story about evolution is Australia. Okay. So here's Australia. Australia has way more marsupials, or mammals with pouches, than anywhere else in the world. There are marsupials in other parts of the world aside from Australia, but not nearly as many, right? So my personal favorite marsupial in North America is a possum, right? Opossums are marsupials. They have a pouch. They, have their babies in there when they're teeny, teeny, tiny.
Hold up, what?
What?
Possums have pouches?
Yeah. They do.
Not okay.
They do. Once their babies get big enough, because they have so many babies, they can't keep them in their pouch anymore. So then the babies like hold on to their backs like little ticks. They're not ticks, when they're about this big, when they're, super adorable. Baby possums are the cutest things ever. I know everybody's afraid of possums and think they're ugly, but they are really adorable, amazing animals. So that's an example of a marsupial that we have, right? Okay. So, all right. Why? Why are there so many marsupials in Australia and not so many in other places? Well, it has to do with the moving of you guys are funny in the chat, sorry. Right, so we have to look at the movement of continents. Continental drift, this is something that I hope you're somewhat familiar with, primarily because this is really an important sort of real life concern, everyday concern here in Southern California, because we went on a trip recently where we drove right over here, right? The San Andreas Fault is the connection between two plates, the North American plate and the Pacific plate, right? And so these plates of the Earth's crust move around. This is not a geology class, so we're not going to get into like the details of why, but they move, and it's slow, right? But they run into each other. And when they run into each other, sometimes they make mountains, sometimes one plate goes underneath another one. But in any case, that produces a lot of movement, like earthquakes and other types of activity like volcanoes and things. Okay, there are other places, right? So in this case where we live, our plates are being shoved together. Okay, there are other places where the plates are separating, and so new crust material, essentially magma, right, is coming up and making new crust in the places where the plates are separating. And so it's a slow process, but it is a measurable process. And so we can look at the current shape of the continents and know how they're moving to, kind of look back in time, Pangea, right? To look back in time and see how the continents have moved, right? So essentially what has happened is over Earth's history, we're not going to worry about dates, but over Earth's history, what has a tendency to happen is the continents all kind of like clump together to make a really big continent. Pangea is one of them, is what we called one time. Is what we called that large clump once upon a time. Okay. And then they separate and then they clump and then they separate and then they clump. Right. So, okay. So that's what happens. And you can see, I mean, anybody, right? A kid who's used to doing puzzles would tell you, hey, look, this kind of looks like it fits. That kind of fits over there. And like this kind of fits. That's like a puzzle piece. Yeah. That's because once upon a time they were connected. Right. And then they're splitting apart. Okay, so why does this matter? Well, the reason that it matters to our story is that if we look at the most recent clumping and we look at how the different continents separated, right? So the yellow ones are the southern, are what are going to become the southern hemisphere continents. The not yellow ones are the northern hemisphere continents, kind of. Right, but there's some weirdness happening because there's a certain amount of like squishing together and separating. And so what you'll notice is that this little landmass here, which is to become, wait, that is India. This little landmass here, sorry, I got confused for a second. So we see that this thing breaks into what will become Antarctica, parts of South America, parts of Africa, India, and then this kind of edge of Antarctica here, that's Australia, right? And so this whole area is separated real early from the rest of these, okay? And that time period coincides really well with the evolutionary history of mammals, right? So the first mammals to develop were similar to modern monotremes. They had eggs. Duck-billed platypus is a monotreme, right? So they're mammals because they produce milk and other reasons, but they lay eggs. Okay, so marsupials are kind of the middle type of mammal where they don't lay eggs anymore, but they have a really extended external gestation kind of thing in a pouch, right? So their babies are born really underdeveloped, and then they spend a lot of time, they finish cooking basically in the pouch. Okay, and then eutherian mammals, that's us, right? and all of what we think of as, your typical mammals, we have longer gestation, so that when our babies are born, they don't need to be like, housed in a pouch to finish developing, because they're basically ready for the outside, more or less. Okay. Being a eutherian mammal is a lot better by all accounts than being a marsupial, because marsupials, their babies are born when they're not done, they're not ready, right? And so there's a lot of risk associated with that. And so it is not very common anymore, because eutherian mammals are pretty much better adapted to life than most marsupials are. So why do we still have so many marsupials in Australia? Well, Australia broke off before eutherian mammals evolved, right? So Australia broke off and sort of went over here, right? And it's got its marsupials, and that's cool, right? And Antarctica is so cold that, it's tough there anyway. So, not a lot going on there. And eventually there are some, but, some mammals, but that's, kind of a different issue. Okay. And so it broke off, and a sizable distance, right? A sizable distance from the other continents before eutherian mammals evolved, right? And so essentially, Australia has the, marsupial equivalent of all of these placental mammals that exist in other parts of the world, right? The big problem for Australia's marsupials is that when Europeans came to Australia, right, they brought with them all kinds of things. They brought rats and rabbits and dogs and cats and, right, and all kinds of placental mammals, eutherian mammals. And that has been really bad news for the marsupials in Australia. So the reason this picture is black and white is because the Tasmanian wolf was thought to go extinct in the, I can't remember if it's the 50s or the 60s. But they haven't found any. There was one that was in a zoo. At some point there were a few that were in zoos, but the last one that was in a zoo died quite a few decades ago. And they haven't seen them in the wild since then, right? And so what's happening is that lots of these marsupials are in trouble or going extinct because they can't compete with placental mammals, because being a placental mammal is just better. Okay. But it's still a cool story to help us understand this evolutionary process, right? So the fact that Australia has such different mammals than other parts of the world tells us that, around the time that Australia broke off of Antarctica I don't think it was called Antarctica at the time, but you know what I'm saying, right? Broke off was around the time that was before eutherian mammals, placental mammals, I should say. Either the names are both accurate. Develop. Okay, so that's biogeography. So biogeography is basically kind of looking at the history, looking at what's around now to infer history. Okay. So another example of biogeography is there are a type of fish called lungfishes, right? And lungfishes are found living only in the southern hemisphere, right? So South America, Africa, a little population in Australia. But they were once upon a time found in lots of different places, right? And so, once again, this kind of combination of looking at how the different plates move and where the fossils are found kind of tells you a story about when something evolved, right? And how that is connected to the movement of plates. Okay. So that's biogeography. And there's where this is not the exact area of the fault that we were driving on, but there it is, the San Andreas Fault. Right. So we drove right across that, you guys, on our trip, week 2. Okay. So that was our second piece of evidence. So we have the fossil record, we have biogeography. Both of those things tell us a story about the history of life, right? And how things came to be where they are. Okay. Any questions about biogeography, or are we okay with that? Okay, check the time. Oh, snap. I gotta pick it up. All right. Third line of evidence. Okay. So now we're on number 3. Third line of evidence. No. Is homologous structures. So this, I'm actually going to end the show for just a second. Yes, keep them, whatever, I don't care. Because I want to show you guys, there's a really great, where did I put it in Canvas? Nope. What do I get for being disorganized? Come on, close, please. Thank you. Thank you. Okay, I don't know where I put it. I'll find it and then I'll like add it to this lecture because it might not. I might be. It's also entirely possible I'm getting this mixed up with 62 because that happens. So I'll find it and I'll post it in the thing for the page for today's lecture in Canvas. Okay. So our third line of evidence for evolution is homologous structures. So have we talked about this? Have we talked about this? We started to talk about it. Actually, no, I'm not going to do that there. I'm going to do it this way. Have we talked about homologous structures versus analogous structures? I think we might have talked about it briefly yesterday. Right, so I'm gonna, can I insert a white screen? So homologous. There's my gap in my screen that doesn't work. So homologous structures due to, look at the chat. Yeah, I know, I talk about it in all my classes, so we might have talked about it in 62. Homologous structures are due to common ancestry, whereas analogous structures are similar in function sorry about the super messy writing. It's the best I can do. Okay, so homologous structures are structures that are similar in their structure due to common ancestry, right? Whereas analogous structures are similar in function, but not necessarily in structure. Not necessarily in structure. Okay, and I'm talking about these right now because we have to watch out for them, but homologies are what our third line of evidence is, right? So this is number 3, is our homologous structures or homologies. Okay, so homologous structures, they can be genetic homologies, they can be developmental homologies, they can be structural homologies. Fundamentally, what that all means is there are similarities related to common ancestry. Okay, so let's look at the bottom one here first, right? So when you look at the forelimb, right? So the arm or front leg or wing or flipper or whatever, right? Of typical vertebrates. So here we're looking at a bunch of mammals and a bird too. Why not? Why not throw a bird in there too? Right? You'll notice that all of these animals have a similar arrangement of bones, even though the function of these limbs is dramatically different, right? So we all have a big bone that's in blue. It's called the humerus, but you don't need to know that. Radius and the ulna. So that's a pair of two kind of smaller bones. Beautiful, right? Kind of thinner bones. And then we have a bunch of little bones, and then some more little bones, and then some more little bones, right? And so we all have this basic arrangement of bones. And if we were in the lab, I would be like pointing to everything around us that is so beautiful, examples of homology, but I can't do that when we're all at home. All right. And so even though these limbs are all used differently, right? Seals use theirs for, kind of steering where they're swimming, but also for kind of like pushing themselves around, right? When they're on land. Horses use theirs for walking and jumping. And we use ours for all kinds of other things, not so much related to locomotion though, right? We're not using our forelimbs to walk around. We're using it to carry stuff and write and pick our noses and, whatever else. Okay. Birds and bats are both using their forelimb to fly, but even then they do it differently, right? So birds, the surface area that they use as a surface for their aerodynamic nature is their feathers, right? Well, bats don't have feathers because they're mammals, not birds. So they're using a sheet of skin, but that sheet of skin needs to be held open so that it's aerodynamic. And so bats are using their fingers for that, right? So even though these functions are totally different, this similar arrangement of bones that's been sort of modified for different purposes, right? That was very teleological, by the way, right? It's not like it was modified on purpose. It was modified by natural selection, right? To end up being used for different purposes. You know what I'm saying? Okay. Right. I say teleological things all the time. I love it when you guys catch me too. Okay. Right. That's an indicator that shows these organisms share a common ancestor, because even though they have such very different functions for those body parts, they really, structurally are very, very similar. Okay. This can be applied to genes too, if we look at similar genes, right? So are genes the same when we're comparing one group of organisms to another? And when genes are closely related, they often are similar, okay? Or more similar than if there's more different. Anyway, you know what I mean, okay? The other thing is developmental homology or embryology, comparative embryology. And so comparative embryology, and those of you who have already taken 62 are starting to like twitch right now because you're like, ew, embryology, I hated that. I don't even want to think about that. But this is going to be in the chat. No, not this again. Exactly, Elle, exactly. But we're not going to talk about it that much right now. All we're going to say is that when you look at the development of embryos, you often see structures that are found in organisms that ultimately don't stick around in the adult form of that organism, but they are there as a result of common ancestry. So a really kind of classic example that people always talk about are pharyngeal pouches. So do I like this picture better? I don't know. Either way, pharyngeal pouches are these like lumpy things on the side of the neck. Okay. And what we're looking at here is we're looking at an embryo of a chicken, a human, and a house cat. All of us have pharyngeal pouches. What do those pharyngeal pouches become? Well, since humans and house cats are both mammals, in us, it becomes part of our jaw, our middle ear, kind of that kind of stuff. In a chick, right, it becomes a different part of their jaw, right? And some other things kind of associated with that. In fish, it becomes gills, right? So we have these structures that are very similar to other animals that we're related to, even though they essentially kind of disappear as adults. Okay. Same thing with the tail, right? So all vertebrates have a tail when they develop. Some keep their tail, some lose their tail. Look at how long this tail is in this human embryo, right? Once upon a time, you had a rocking tail, right? It was probably about half the length of the rest of your body, right? Do you have that now? No, you do not. And if you do, you are doing an amazing job of hiding it, because I don't know. Okay. Right. Whereas cats keep their tail, right? Chickens lose most of their tail, but they have like a little nubbin, little butt nubbin tail. Okay. So right. So comparative embryology is essentially just homology of embryos. Okay. I'm gonna write that down. Nope. Homology in embryos. Okay. Different organisms, same structure. So in this image, they're calling it a gill pouch, but right, pharyngeal pouch, and then the tail, right? So in our shark, the gill pouches become gills and the tail stays a tail. In turtles, the tail stays a tail. They don't have gills though, right? Anyway, okay. All right. You have to be careful though, because there are some structures that are analogous, meaning that they have the same function. They might be misleading. They make you think that they are potentially in homology, but they are not. So this example is kind of a really clear example. Others are not so clear. So here we're looking at a bat, a puffin, and a housefly. So all three of these organisms have wings, right? Which ones are analogous and which ones are homologous? So which ones of these are similar as a result of common ancestry, similar in structure as a result of common ancestry? We looked at the example before. Ding, ding, ding.
Wait, wouldn't it be A and B?
Right. Yeah. So wings are homologous. Okay. All right. Now, we know that because they're both bony. They both have muscles and skin, right? So even though they're not identical, their main structures are the same, right? Are fundamentally the same. Okay. What about if we compare our puffin wing and our fly wing? Well, our puffin wing is made of bones and muscle tissue and skin and feathers. And our fly wing is basically just a little flappy extension of the exoskeleton. Totally different function, right? It's not different function because they both use flying. Sorry, let me say that again. Totally different structures, right? They're not coming from similar origins. They're not similar in their structure. So while superficially they're used for the same function, right? We would say that a puffin wing and a fly wing are analogous structures instead of homologous structures, right? So analogous wings between those two. Okay. Sometimes it's tricky because if you don't know what you're looking at, you can make a mistake, right? So here we're looking at a couple of different plants, and it's like, okay, these two plants have leaves and they have sharp, pokey things. Are those sharp, pokey things homologous or analogous? And to the casual observer, you might think, oh, those are probably homologous. Those probably are a result of common ancestry. The fact that it's like pokey plants and pokey plants are similar to each other. But actually, they're not, right? So in this case, the pokey structures are made from different structures. So these pokey structures are extensions of the epidermis of the plant. So they're like little outgrowths of the plant's skin, for lack of a better word. Whereas over here, these pokey things are basically modified leaves, right? So some of the leaves, not all of the leaves clearly, are modified into these sharp, stabby things to provide protection. So while the function of the stabby things in both of these plants is the same, the function is the same, they do not come from the same, tissue origin, right? And so what we end up with is analogous structures. Okay. So analogous structures, how did I end up? Wow. Okay. So you just have to be aware that those exist, right? And that they're somewhat problematic. Okay. But an interesting thing about analogous structures is analogous structures are a result of convergent evolution. And so this is something that I've been like saying to you guys. I say it in 62 a lot. I think I might have said it to you guys once or twice, but convergent evolution basically is when a function evolves more than once. Independently in separate groups. Independent in different groups. Right? So essentially these different organisms are converging onto the same function, right? And the way that I love to describe this when I'm talking about this is I always say that when you see something that evolves more than once completely independently, that probably tells you that that function is super useful, right? It's got to be. If it's something that I got to move, you guys were in my pictures of you were in the way. Super useful, right? So we see flight in at least 3 different independent groups: insects, birds, and mammals, right? And it evolved independently in all of those groups, right? But it shouldn't be surprising to us that it did, because it is such an incredibly useful trait, right? And the way that that function evolved in those groups is really different, right? So we were talking about how insects are using their nubbins originally for heat, and then it ultimately became wings, right? So the origin of that kind of a wing and how that went down is very different from the origin of like a bird wing, where it's like, initially they were kind of useful for climbing and gliding, and then over time they change. Yeah. So convergent evolution, right? When a function evolves more than once independently in different groups, right? That's convergent evolution. And typically we're talking about things that are really, really handy. And so we see this in this kind of characteristic in a lot of different organisms. Okay. And here's an example of that very thing, right? So when we look at, comparatively Australian marsupials and their placental counterparts, right? This is kind of an interesting story of biogeography and convergent evolution, because even though they are different organisms living in different places, they have similar functions in the ecosystem, right? So a giant anteater does the same kind of stuff as a numbat, right? A gray squirrel has a similar kind of lifestyle as a sugar glider, right? And before they went extinct, Tasmanian wolf had the same kind of job in the ecosystem as a gray wolf, right? So that's kind of an interesting, story of looking at both of those things together. Okay. All right. What do I want to say? Yeah, convergent evolution, right? Okay. And then this, one might argue, isn't even really a separate. I'm going to do 3 or 4, right? Because when we talk about genetic similarities, essentially we're just talking about homologies that are genetic rather than structural, right? So genetic similarities. You might consider it a 4th line of evidence of evolution, or you might just say, nah, it's still a homology. It's just homology of genes instead of homology of, body parts, right? So I don't know how I feel about calling it a separate thing or not, but you guys can decide. I don't care, as long as you understand that it's how it's related to the whole thing. Okay. So this little graphic shows how much similarity, and this can be genes and also so actual like DNA sequences, but also amino acids in a protein, right? So when we're talking about genetic similarities, we can be looking at DNA. Similarities in DNA or proteins, because ultimately those two things are connected, of course, right? So this is looking at the sequence of amino acids in a certain portion of hemoglobin, right? And so it's using humans as kind of like the baseline, and we're seeing how similar it is to different species, right? So one would say that humans and a rhesus monkey are more closely related because this particular area of hemoglobin only has 8 differences in the amino acids when you compare a human with a rhesus monkey, whereas when you compare a human to a bird, it's 45 differences in the amino acids, right? So yeah, okay, so it's it's basically another way of sort of making a family tree, but using DNA sequences or using amino acid sequences, right? And we can actually make phylogenetic trees. We haven't talked much about those yet in this class, and we're not really going to, because I really lean into this real heavily in 62. So we're probably not going to get into it too much in here, and that's okay. Maybe a little bit tangentially. Okay. So yeah. Okay. So we can look at relationships between different organisms based on DNA sequences and similarities. Okay. So, right. So what does this diagram tell us? Well, what this diagram tells us is it tells us that for the particular DNA sequence that they were looking at, right, in this particular, when they made this little table here, right, the similarity between humans and chimpanzees was around only 1.7% difference, right? So 98.3% the same, right? And then they compared the DNA between a chimp and a gorilla, and it's 2.2 ish, and then chimp to orang, and then chimp to gibbon. Yes. Okay. And then what they did is they said, based on what we know about mutation rates, we probably assume that the different organisms on the table, right, we can sort of draw a line and say, all right, the point where these two species shared a common ancestor is about here, right? So based on what we know about how rapidly DNA seems to mutate, right, we can guesstimate that chimpanzees and humans shared a common ancestor. What is that? Probably like 6.5 million years ago. Yeah. Okay. And when you use that piece of evidence along with all of this other stuff, right, looking at homologous structures, looking at the fossil record, blah, 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 it makes a pretty complete picture. Okay. So all of this to say that we have a lot of tools that we can use to ask ourselves, essentially, right, to ask ourselves is, how did different organisms evolve, right? And so we don't use only one of these types of evidence, right? We use some in certain circumstances, others in other circumstances, but generally we look at them kind of all kind of collectively to see the whole picture. Okay. So for example, we might look at genetic similarities plus also biogeography. Where are all the fossils? Where have the fossils been found? Right? What areas? And, also let's, see if there are fossils, right? So all of this kind of together is our evidence that evolution has happened. Okay? All right. Questions? I'm looking at my clock here. How are we doing? We're not going to do too much of this because we're just not. We're not. We're going to skip it. We're just going to skip it entirely. Okay. We're going to deal with this. Woo! It squawked at me. Okay. So I'm going to stop sharing. All right. At least for a minute. And I'm going to, what am I going to do? I'm going to go and I'm going to find my outline. And I am going to, am I going to update my outline? Where is it? Here we go. And we're going to update it, and then I'm going to save it on Canvas. And I'm doing this with you guys here so that you can take a minute to think if you have other questions about things that you need to prepare for the. So this, as it turns out, is the only thing that we're going to end up skipping. And I'm not, in fact, I'm not going to do it on purpose because we're not going to save it for next time. We're just not going to do it, really. Okay. Save as. Okay, let me share my screen so you guys can see what I just did. Right, so. Right, so I just took these off. Okay. All right. You guys have any questions about what to expect? I will post this on updated one on Canvas.
So the red means we're never going over this in this class.
Not really.
Thank God. Sorry, I saw by
binomial nomenclature.
Yeah, and got PTSD.
Well, I mean, here's the thing. We are going to talk about it a little bit because in lab reports, it's going to be important to me that you write scientific names correctly. So it will come up in that sense, but it's not going to be like on the exam, right? So we'll talk about a little of this kind of sometimes, but not in a very formal way. Okay, I'll just put it that way. Okay.
Oh, thank God.
All right. Okay. Any other questions?
I have a question. So on the exam, will there be like pictures of two different like species, and then do we have to like say if it's like analogous or like homologous or no?
That would be amazing, but I would only do that if there was sufficient information for somebody without a lot of detailed knowledge about something to be able to answer that. Oh, if that makes sense.
Mm-hmm.
Yeah.
Yeah. Thank you.
You know, but here's the thing, you guys, right? Like the classroom that we have, that we use, has so many examples of homologous structures. And for that matter, even has some examples of analogous structures, but mostly homologous structures. It's like, how could we not? How could we not ask questions like that? Come on. Anyway. Okay. All right. Anything else?
Just a little comment. If you're in the Discord, I'll be hosting a study session sometime tonight, maybe. So please join. I don't want to be alone.
That's how I feel every time I Zoom L. I'm like, please come. I don't want to be more stuff. Which, speaking of, I have office hours now. So if anybody has anything you want to talk about, like one-on-one, you know, you can come back at 11. But I do want to just end our recording. So in fact, I'm going to do that now. I'm going to stop recording now.